So I was going to say a little bit about the idea of treating aging or anti-aging treatments. And this is a complex topic. Let me say, first of all, I think I should say this, which is that I'm all for that. I think we want that. And the reason why is because aging is fundamentally a horrible thing. In many ways, I think, it's part of the human condition, which is almost maddening, and somewhat similar. I say it to the students sometimes, to a situation where somebody comes to you and they tell you that, okay, in 20 years, some men are going to come and knock on your door in the middle of the night. They're going to take you away and they're going to torture you for about 2 years until you die. And that's pretty much our condition as human beings. And that's what happened to both my parents. I watched that happen to them as they were killed by diseases of aging. So aging is a horrendous thing. So in terms of the idea of treating aging, there are problems, I think, with thinking about what do you mean by treating aging? What is a treatment for aging? So I've argued that you can't really make a distinction between aging and aging-related disease, although sometimes people do. So a treatment for aging is a treatment for aging-related disease as well. So does that mean that having a coronary bypass operation or having a hip replacement operation, that's a treatment for aging? Is that an anti-aging treatment? That's not usually what people mean. I've also argued that there probably isn't a central mechanism of aging, although some of the work on the short-lived animal models like C. elegans gave the impression that was the case. I think this was illusory, actually. So if there isn't a central mechanism of aging that you treat, and if there's not a difference between aging-related diseases and aging itself, what is an anti-aging treatment? What does it really mean? What I would propose as a sort of solution to this problem is actually a new definition of anti-aging treatment, which is to say that really, when you talk about anti-aging treatment, you're talking about treatments that are preventative. And that's partly to distinguish them from simply everything to do with any aging-related disease. It also kind of uses the anti-aging. You're essentially preventing them from happening in the first place. And it also acknowledges the fact that in the laboratory, this is what we can do. This is what we've learned. One of the things we've learned in the laboratory is that you can intervene and prevent, or at least decelerate, the appearance of aging-related diseases. And this is very strong across the animal models. But by that definition, that would mean that, for example, things that affect even prevent one aspect of aging would be anti-aging treatments. So, for example, sunscreen to prevent UV radiation damage of skin. So that's part of aging. And you can use sunblock, and sunblock will prevent. It will delay and protect against that aspect of aging. So that's an anti-aging treatment. Or even if you go to the hygienist and you have the plaque taken away from your top of your teeth, and that prevents aging of your teeth, that's a preventative treatment of an aspect of aging. So that's an anti-aging treatment. In terms of the older notion of anti-aging treatment is something against the whole of aging. This is really interesting topic. So in C. elegans, you can manipulate individual genes in C. elegans and see extraordinary increases in lifespan. You can double and triple and quadruple their lifespan. Even one group increased them up to tenfold. And an implication of that, this is what drew me into this field, was that there's some sort of extraordinary core processes of aging which underlie the whole of aging. And if you could understand that, you could do extraordinary things with human beings. You could have people living, you could slow aging right down and just stave off diseases of aging for many, many decades, wonderful. But I think that notion was also consistent with some of the theories of aging that were predominant in the 90s, particularly like the oxidative damage theory. So there was a notion, people expressed the idea that if you could enhance maintenance processes within organisms, you could essentially control completely the accumulation of damage, and you could extend lifespan dramatically. So this is what people kind of thought they were doing. But I think more recently, it's become clear that the higher up the sort of the evolutionary ladder you go, from worms up to flies to mice to rhesus monkeys to humans, the less plasticity there seems to be with the less of a sort of innate plasticity there seems to be in aging. So one of the things which I think has been a big shock over the last years was the results of tests of an intervention called dietary restriction on rhesus monkeys. So this is an intervention which in rodents and many other short-lived animals slows aging down dramatically. So in mice and rats, you get 50% increase in lifespan, suppression of a whole range of aging-related diseases. And this was tested on rhesus monkeys, and the results were really quite disappointing. At most, the effects that were seen, the small effects, look as if they were rather likely to be rescue of harmful effects of overeating, which is not what you see in the rodents. It's something quite different. So I think that at the moment, it doesn't seem likely that there's any sort of magical plasticity that you can tap into in the way that you can in, for example, in C. elegans. However, things change very rapidly within the aging field, and that's one of the features of this field, which is rather marvelous if you're working in it. It's so unstable. Theories rise and fall very, very quickly. And something that's happened very recently, which is just incredible, is the development of recent findings in rodents looking at a particular type of cell called a senescent cell. This is a very particular type of cell that accumulates with age and has been shown to contribute to the generation of a whole range of different aging-related diseases, from multiple forms of cancer to cataracts to cardiovascular disease, many other kinds of pathology. And experimentally in the mouse, if you clear these cells, which you can do through complex genetic interventions, and you can now do it with a class of drugs that are called senolytic drugs, you get these remarkable effects on health. You get essentially a broad spectrum protection against diseases of aging. But the mice still get the diseases, they just get them later. So it isn't affecting the entire aging process, but what it is doing, it seems to be affecting, as it were, a cause of senescent multimorbidity. So it's an underlying cause of multiple diseases of aging. So this is something radical and exciting and quite different to the way that conventional medicine is generally looking at diseases of aging, which is to wait until people get the diseases and to study them individually in separate disciplinary silos, in oncology or cardiology or and so on. So this just looks wonderful, but it's in mice. So I think the prospects for this working in humans, personally, I think it looks quite good, but we've been here before many times. So I think I'm not particularly optimistic about interventions that are going to have major effects in terms of adding many years to life or anything based on the technologies that are around at the moment. But I think the possibilities, for example, for senotherapy, these drugs that clear senescent cells, as an example of something that's come out of the experimental biology of aging, is very, very promising at the moment. I mean, I don't want to give the impression that I'm being pessimistic about the aging field. I mean, I think that in a way I feel both pessimistic and optimistic at the moment, because there were a number of elements of the field that looked extremely promising, like this huge plasticity in aging that you see in the short-lived animal models, the oxidative damage theory and the whole damage theory of aging, which as an intervention that was going to have dramatic effects, dietary restriction, which looks quite disappointing. And I think the thing which doesn't seem to be there is this natural plasticity in aging that you see in shorter-lived animals, in humans, at least in terms of the possibility of slowing aging down. And one of the recent results that I think is consistent with that is that the maximum lifespan for human beings worldwide for many, many years, for many decades, was increasing year on year. But it's actually hit a ceiling, it's plateaued. And it did that around about the year 2000. And it's even becoming clear that some of the very long-lived people, the real record holders worldwide, that a number of them may be essentially unreliable evidence. So, for example, it's recently been, a case has been made that a woman who was thought to be the oldest woman in the world, Jeanne Calment, was actually not actually Jeanne Calment, it was actually her daughter, who she switched identities with when her mother died. So on the other hand, I think if we kind of turn things upside down, one can make the case that that there is a plasticity in human aging, but it actually runs the other way, which is that you can accelerate aging in human beings. And that's something that very much seems to happen when people eat too much. So when people become very overweight, it would appear that the aging process is speeded up. So one possibility is that interventions that actually protect against aging in the short-lived animal models actually could be used as ways to prevent accelerated aging in people who are overweight. But in terms of the, optimism, the thing that really makes me optimistic is the possibility of really understanding aging. This is quite aside from what you can do with the knowledge, because that must be possible. And I think over the last years that the basic concepts of aging have changed so much, and they've sort of evolved and transformed so much, that we must be coming close to the point where we have the basic foundation of an understanding of aging. And that has to be the way forward to be able to see what can be done in terms of preventing late life disease and allowing people to live longer, healthier lives in the future.
Hundreds of labs around the world study aging. And one of the latest ideas is that aging might not just be slowable, but actually reversible. And this is something that I thought I'd never see in my lifetime. Now, the reason that I think it's quite remarkable to even talk about reversing aging is that just 20 years ago, the idea of even having an impact on aging with genetic changes or small molecules, drugs, was considered crazy. I got into this field about 20 years ago when I was a kid. And a lot of people advised me not to go into aging. It's bad science. It's the backwater of biology. And I just thought it was a really interesting thing to study, and that it could have a really big impact on the world if we could do something about aging. I'm doing this not so I can make people or see people live 1,000 years. I don't think that that's going to happen anytime soon. But what I'm doing this for is to be able to have a big impact on human health. So what about reversing aging? Now, one of the reasons that we've been surprised that aging is so reversible, at least in simple organisms, is that a leading dogma in the theory of aging, why we age, is the mutation theory of aging. And that's the idea that our bodies accumulate mutations over time, and that leads to the decline in our body's function. And as you may well know, mutations are largely irreversible. Now, the good news is that maybe it's not the mutations themselves that cause aging, but the cell's overreaction to the DNA damage that occurs as we live. When I was just a young postdoc studying aging in a simple organism, Saccharomyces cerevisiae, which is the technical term for baker's yeast. Now, these are little yeast cells that the world uses to make bread and beer, champagne, for example. And this organism is really interesting and useful because you can change its genes very easily. You can study its lifespan within a week. They only live about a week. They produce about 25 offspring and then they die. And the first thing that I wanted to study when I was doing my postdoctoral research at MIT in the lab of Lenny Guarenti was, why do these cells not live forever? They're relatively simple. They just divide, they split. So why couldn't these be like stem cells? Why couldn't they live forever? Well, it turns out that the problem with these cells, a main problem, is that their DNA becomes unstable. And what happens is on the linear chromosome, they have 16 chromosomes in a haploid cell. The chromosomes pinch off a circle of DNA, and these replicate, and they end up choking this single mother cell. Normally, if a cell was smart, it would divide that split that circle between the mother and the daughter cell, and you just have a circle between the mother and the daughter, and that would be no big deal. But the problem is that the cells don't know how, or don't efficiently put this circle into the offspring. They accumulate it, they keep it in the cell, and the cell thinks this little circle of DNA is another chromosome, so it replicates that, and you get exponential growth of this circle. And actually within a number of divisions, about 12, 13 divisions, you end up with so many circles of DNA in the thousands that they outnumber the rest of the chromosomes in that cell. And they choke the cell, they titrate, they suck away the proteins, and the cell can't divide, and they die. And we were able to show in that lab that if you delay the circles from forming, or you prevent them from replicating efficiently, the cells can actually live longer, and we could extend lifespan in that organism. And that was one of the first known causes of aging, and one of the first ways to slow down that known cause. Somewhat disappointing was that when we looked for these circles of DNA in our own cells, in mammals, we didn't find many of them. And in fact, we don't think that these circles themselves are the key to immortality in mammals or in ourselves. But what we do think is that there's a principle there that tells us about aging in our bodies, and that leads us to this reversibility of aging. So what we then discovered when we were studying this circle formation was that there were these proteins that tried to suppress the genome instability. There are proteins in our cells as well that go to DNA regions of instability where, for example, there's a break in the chromosomes or the DNA is highly repetitive, that try to stabilize our DNA. And that's true for yeast cells as well. Now, one of these proteins turns out to be really important for this process, and its name is SIR2, SIR2. Now, in our bodies, we have 7 of these SIR2 genes. We call them the SIR2ans, and we think they play a very similar role in humans as they do in yeast. That's maintaining the stability of the chromosomes that are apt to recombine and form breaks and circles. Well, one of the most amazing things we discovered was that these proteins that are normally bound at the ends of chromosomes, the telomeres, which many people will know is one cause of aging, the loss of these repeats. They're bound at various regions of the chromosome, and they control whether genes are turned on or off. SIR proteins in yeast are very good, and their main purpose is to keep genes switched off that should not be coming on unless the cell is stressed. Now, what the problem is that when you get this genome instability in the broken DNA and the circles, the SIR proteins have this trigger that the cell says, hey, I know you're busy over here silencing, controlling these genes over here, but we need you really now for this genome instability. These circles are popping out, the chromosomes are breaking, get over here. So the cell recruits them in a very active mechanism, pulls them to the region of where there's a problem, and the SIR proteins help repair the DNA. They bind to it, they make it fold up correctly, and the DNA break is repaired more efficiently. The problem, though, is that if they don't go back to where they came from, these genes that were once silenced and controlled by the SIR proteins are now expressed. They come on, you get a stress response, which is okay in the short term, but in the long run, you end up with a cell that has a permanent chronic stress response, which is not good for a yeast cell, and it's not good for a human being either. And in fact, we now know that chronic stress is a major cause of aging in many different organisms, from yeast to humans. So really what I'm saying in essence is, in yeast we learned that DNA damage, DNA instability, chromosomes breaking, distracts these proteins from what they should be doing, which is controlling how genes are expressed. During the distraction, these genes come on and the cell has this stress response. Now in yeast what happens is, one example is that SIR proteins normally silence genes that control mating. Yeast cells mate, they need to find the opposite sex. And SIR proteins tell the cell by suppressing certain genes, whether it's a male or a female, A or an alpha cell. When the SIR proteins are distracted, helping repair DNA, you get these sex genes coming on, and the cell doesn't know if it's male or female, A or an alpha, and it actually becomes very confused, and it can no longer mate. It becomes sterile, and that's a hallmark of yeast aging, is sterility. And what we found was that if we could prevent this genome instability, or actually put more SIR genes into the cell, we can put in more genes quite easily, we could do two really important things. One was you could repair the chromosomes better, because there's more SIR proteins to go around, and these silent genes, these mating type genes that cause sterility, stayed silent, and the cells didn't become sterile until much later. We think that the same process occurs in our own cells as we age. What we and others have found is that we do develop breaks and chromosome instability during aging, particularly at repeated DNA. So when there's one gene after another like this, or not just genes, just repeats, the chromosomes are very apt to crossing over, as we call it, recombining, breaking. And when this happens, the SIR proteins in our bodies, they move to the break, and we know that they help repair. SIR proteins in our bodies also control which genes are turned on or off. And we think that when there's breaks in our own chromosomes, that these SIR proteins are moving away from where they should be. And one of the tests of that was to see if we create DNA breaks in our cells, in culture, do the SIR proteins move to the breaks? Yes, they do. And do these genes that are normally controlled by SIR proteins get expressed? Yes, they do. And another interesting thing was that the genes that come on when you have breaks were very similar, had an overlap with the genes that come on during aging in a mouse. And we think that this may be also true for our bodies as well. So what's this got to do with the reversibility of aging? Well, if it's true that it's not the mutations and the breaks in the DNA that cause aging, but the cell's reaction to that, what we should be able to do is to compensate for that reaction. One thing we could do would be to put in more SIR genes, and so the genes make more SIR proteins. So instead of having them all distracted, we could have enough proteins to go around so we could control those genes and have the repair process go on at the same time, and keep those genes silenced when they should. The second thing we could do is to make these proteins have more activity. We now know that if we put in simple molecules, maybe a drug one day, we could make these proteins hyperactive so they can do their job at the broken DNA, and they could also keep the genes controlled. Without these molecules, what happens in our bodies, we think, is that over time, these genes come on and we're left with a chronic stress response. And this stress response leads to the decline of our bodies and aging. So really, what does this mean? Just to sum up, is that if it's not the mutations, and it's the changes in proteins, how they move around the cell, this should be reversible. And we're already seeing signs from work that we and others are doing that we can restore the function of these proteins and not just delay aging, but reverse it as well. This is leading to a whole new field called epigenetics, where we can change how the chromosomes are read, how the chromosomes are wrapped up in the cell, and take that back to a more youthful state. And if we can succeed in doing that, we have a great chance of being able to not just delay aging, but put it off and even possibly reverse it one day so that we can lead much healthier and longer lives.
There are different definitions of what human aging is, what aging is. And probably one of the most convenient is the statistical definition. If we look at population of individuals, they die. Living things tend to die. The question is, how do they die? If they're dying because of random events, like in human population, they may be struck by cars, or they may die because of disease. The typical dynamics of such population will look like the curve which goes down like this. Actually, it's an exponent. So this shows how many individuals are still alive at a certain point. So 100% here, and then 0 here. So this is a typical curve of non-aging population. Individuals are dying with the same pace through the time. The hallmark of aging is a different type of curve. Aging is the situation where the rate of dying increases with time. So a typical aging population will die something like this. There's almost no deaths in the beginning. The number of individuals is at 100%, and then at a certain point the rate of dying increases and goes to 0 eventually. Interestingly, our human society became really aging only recently. It used to be that the death curve of human population looked something like that, something intermediate between the curve of aging and non-aging population. And actually what happened, it was there are a lot of deaths because of early reasons such as disease or wars, huge child mortality. And then if person did live long enough, he did actually age. So you could see a little tail here which looked like this, which looks like an aging population. The history of human society in the last century and a half was actually the conversion of this non-aging type of population to aging type of population. But from the point of view of somebody who is studying aging, is that not only did this curve become what is called rectangular, but in the last 50 years, the dynamics switched from what was called rectangulization, this into this, to the actual movement of the whole curve towards older age. What this means is that we switched from fixing early deaths, or mostly fixing early deaths, into fixing diseases of aging. So what we see now in the human population is actually curing of aging. And we can look at this problem from different points of view. Probably one of the most spectacular ways of presenting human longevity is so-called maximum life expectancy plot. What is done is we look around the world for the country where life expectancy at birth is the longest at that point. And then we plot these life expectancies against time. So this plot is available for 150 years approximately. And the most amazing thing about this plot is that it's a straight line. It goes straight for 150 years without fail. Actually, different countries become the top countries in terms of life expectancy. First, it was New Zealand for very many years. Then it became Sweden. For the last 2 years, it was Japan, and now Japan was surpassed by Macau. Macau is a little region in China, which previously was Portuguese colony, and there are about 500,000 people there, so it's a representative group, and they are now the longest-lived group of people on Earth. Perhaps most amazing thing about this line is that it apparently was very resistant to great discoveries in the medicine. Like, for example, one of the huge factors of decreasing mortality was washing hands. When physicians started washing their hands, the child and mother mortality went down humongously. But we don't see anything on that curve. It still goes straight. Antibiotics were discovered, which of course saved lots of lives. But again, at the time where antibiotics were discovered, we don't see any difference in that straight line. About 50 years ago, we switched to the type of aging dynamics. From fixing early mortality, we started to fix late age mortality. Again, the curve goes straight as if nothing happened. So probably the reason why this curve goes straight and linearly is not actually what technological advances will make, but probably more some economical factors or maybe psychological factors, and they actually keep this curve. For example, when antibiotics were discovered, it's the rate with which they were introduced into healthcare which controlled their impact. So basically, our discoveries have to be reduced to practice, and that takes some time. So this becomes a very complex picture. But given that, what do we expect in future? And do we expect that certain age-related interventions will significantly increase our lifespan? And most likely not. Most likely this curve will keep going up and up as it used to be before. So far, we don't see any saturation. Actually, this observation was done about almost 15 years ago. This curve was published. Since then, it keeps going up just in the same pace. So it was a good prediction, and it was tested by time. We don't know how long it will go. Maybe there will be some limit to this increase of our longevity. But so far, nothing like that. What does it tell us about the way we age? As healthcare improves, as we keep fixing different problems which affect our health, some time ago, the major killer was cardiovascular disease. Even before that, bacterial infections. Then bacterial infections were contained by antibiotics. Now the main killer became cardiovascular disease. As our age or life expectancy increases, other factors come into play. Now cancer increasingly becomes a more important factor. And as we keep fighting with cancer, very old people are struck by dementia. Alzheimer's dementia, they're struck by sarcopenia, loss of muscle mass, osteoporosis. Actually, infections come back. Infections are huge problems in nursing homes and geriatric units. So what we see now is that the landscape of our diseases in the old age becomes increasingly complex. Now an old patient will take pills from infection, from osteoporosis, from memory loss. So the management of our health becomes more and more complex. So we are reacting to that by making medicine more and more sophisticated technologically and scientifically saturated. It looks like great discoveries become more and more difficult to implement in healthcare. So that is one of the other reasons why we think that there will be no significant increases in lifespan in the nearest future, even though in the making there are lots of great ideas such as stem cell therapies, personalized medicine, when we will treat every person according to what is in his genome. And of course, if we don't keep investing in science and medicine, we will not be able to keep the pace which we have kept for 150 years, and hopefully we will.
Now, the prospect of living longer has enticed humans probably since we were first conscious. And for that reason, there's a lot of skepticism that we'll be able to achieve this goal, because so many people have failed until, even till now. We have very little way of improving our health or living longer. So why am I optimistic that one day we will live much, much longer and healthier? Well, one main reason is that in other organisms, it's turned out to be rather simple to extend the lifespan of those organisms. And I really don't think that we're that special when it comes to life forms on the planet. Going back about 25 years, the idea that you could do something about aging and slow it down was considered crazy by most people, and I would say today still that's a standard view. Now, the view that gets in the way of that is that our bodies are much like cars. We're machines that are extremely complicated. Of course, we're much more complicated than a car, but we still have this view, largely, that our bodies are machines that wear out over time and break down, and this is what causes aging. Now, if that were the only truth to aging and to biology, then of course we would have no chance of slowing down this process. Because let's say I came up with a pill or someone developed a drug that could keep part of our body healthier for longer. Well, some other part would still break down, and that's actually how we practice medicine today. We prescribe a drug for cardiovascular disease, but this does nothing for Alzheimer's. And this is the real problem that we face. Now, by that car analogy, turns out 25 years ago, there was a number of discoveries in the late 80s, early 90s, that said that maybe there's something more to this idea than just a car breaking down. What we now believe is that our bodies are more like, if you want to use that analogy, we are like cars, but our bodies have inbuilt repair mechanisms that can be brought to life with molecules or genetic manipulations. So instead of just thinking we wear out over time, we actually have inbuilt repair women and men that are working to keep us healthy. So what that means is if we could find a genetic way or a pharmacological way to get these repair people up earlier in the morning or to work harder, we would have a way of keeping our automobile, our bodies, healthier for much longer. Okay, so that now means that it's at least feasible that we could have an elixir of youth, something that could make us live a bit longer. So why am I so optimistic that we could live much longer one day? You know, not in my lifetime, I don't think in most people's lifetime, but one day. The reason is that a number of labs around the world have made discoveries that single gene mutations, just changing one gene, in some cases just one base in a sequence, is enough to extend lifespan dramatically. The best example I can give is work from labs such as the Kenyon lab, Ruvkin lab, Johnson labs. They found that mutating the genes in a simple worm, a little nematode worm called C. elegans or Caenorhabditis elegans, Caenorhabditis elegans, that this little worm that's about 1 millimeter long can live twice as long with just one mutation in its genome. That gene, those genes in that pathway turned out to be related to the insulin signaling pathways that exist in our bodies as well. And there's a lot of work that's going on to try and understand how that pathway in our bodies could also be manipulated to make us live longer. And there's increasing evidence actually from centenarian studies, these are people that in their families tend to live over 100 years, that this same pathway in the worm is also important for making us live long as well. But this one genetic change, or this one genetic pathway that I'm talking about, isn't the only one that's important for lifespan in the worm, and probably isn't the only one important for lifespan in humans as well. We now know that there are dozens of important pathways that we've discovered in these simple organisms like worms and fruit flies, and even now in monkeys, that are able to delay aging quite dramatically. So how do we think that these genes work? I'm often asked, aging is really complicated. How can these genes extend lifespan? Well, it gets back to that analogy about the car and the repair people. What we think is that these, what we call longevity genes, control these repair people. So instead of our bodies working an average 9-to-5 job, keeping our cells healthy, what these genes do when they're altered is trick the body into thinking that there's an adversity coming or there's not enough food, there's not enough DNA repair, what that does is it tricks the body into mounting this stress response and repairing the cells better. It's as though you make a phone call to the repair shop of the car. You say, the car, it really needs to be repaired. It's smashed. Quick, get everybody out of bed and work harder, when actually everything's perfectly fine. But they polish the car, they make sure everything's working well, and that, we think, would keep our bodies healthier for much longer. So that's actually a really novel discovery, as I say, only in the last 20 years or so, that we've realized that there are inbuilt repair mechanisms in not just our bodies, but all of life. And these genes that I'm telling you about, the insulin signaling and others, are found in all these life forms. Now, another question that's interesting is, why would these genes exist in the first place? And why aren't they switched, if they exist, and we know they exist, and then if so, why aren't they switched on all the time if they're so good for us? Well, what we think is that they evolved to counteract adversity and keep these organisms and ourselves alive for longer when there's not enough food around or there's some other stress. You can imagine back in 2 billion years ago in the primordial soup, when the life forms are swimming around, early life forms, even some of the simplest cells, they needed a way to adapt when the conditions became harsh. Let's say there wasn't enough of a certain molecule available. Well, they developed ways to respond to that stress. And what we found is that these genes that are very ancient are found in all life forms now. And when organisms such as ourselves, or even little worms, they don't get enough food, or they don't get enough vitamins, they don't get enough amino acids, that tricks the body, or tells the body to get into a state of super repair. So why aren't they switched on all the time? Well, you don't want the body to always be in a stress response. That doesn't make sense. There are times when there's plenty of food or nutrients available. Then what you want to do is switch off these pathways and allow the organism to do other things that it needs to do, such as grow, get bigger, multiply, and reproduce, produce offspring, which is, of course, essential for life. And we think that these genes come on and off depending on whether the environment is harsh or not. Now that leads me to my last point, which is what are these genes telling us about a very interesting diet known as calorie restriction? Since actually the early 20th century, it's been known, and you could argue earlier than that, that restricting the amount of food that animals get makes them healthier. And we've known for the last, for millennia, that fasting is healthy, and that's still true today. The first rigorous experiments were done in 1935 by McKay and colleagues, and they in the US, and they found that restricting calories of rats by about 30%, 30 to 40%, greatly increased their health and made them live longer. And this was really a quite dramatic finding, and it's been studied ever since by people who want to understand how to slow down aging. There are people today who are calorie restricting themselves in the hopes that it will make them live longer, though we don't yet know if that's going to happen. But calorie restriction is amazing because it's the most reproducible and robust way to extend lifespan in very different organisms, from these little worms through to monkeys, in fact. So what we think is going on under here, under the hood of the car, is that the diet is tricking the organism into thinking that the conditions are so harsh that it has to constantly repair its cells. These genes that I've talked about, the insulin signaling and these other so-called longevity genes, they respond to the lack of food, and they come on, and these defenses come on. And then the final thing to think about is, what are we doing to our bodies by always having food available in our society? We're very rarely hungry, and fasting for most of us is something we do rarely, if ever. I think we're doing to our bodies is that we're shutting down these ancient defense pathways that would normally keep us healthier. Perhaps one day we will find molecules that we can take as a pill, for example, that will, instead of having to go hungry, to fast all the time, we could pop a pill that would tell the body, or at least trick the body into thinking that it's not getting enough nutrients or enough food, and it would turn on these inbuilt repair systems, these innate defenses that exist in our bodies, and those would keep us healthier for longer. And perhaps when we're in our 90s and 100s, we could still be healthy and play tennis with our great-grandkids and teach them all the wisdom that we've accumulated over the decades. Maybe one day we could live twice as long like that little worm was shown more than 20 years ago. And if we live in a world like that where people live healthy, longer lives, I think it would be a much more interesting and productive world to live in. And actually, I think those people will look back at today with some sort of pity that we only live these short, relatively unproductive lives.
COPD is a disease of the elderly, so the main age of diagnosis of COPD is 60 to 70 years old, and it never occurs before the age of about 45 because the disease takes many years to develop. But there is increasing evidence that this disease may be due to acceleration of the normal aging process in the lung. And this is an important discovery because it's led to an understanding of the molecular pathways that may be involved in accelerated aging. And this in turn is important because it's identifying novel therapeutic targets for the future. So lung function reaches a peak at about the age of 25 years of age and then slowly declines over decades. But it never reaches the point where this decline leads to symptoms in normal people. But if you look at the lungs of elderly people, they do show some of the pathological features that we normally see in COPD. So if you look at people of 90 or so, they have what's called senile emphysema, with large air spaces in the lung parenchyma and inflammatory cells, which are features of COPD-related emphysema. But our hypothesis has been that the acceleration of aging accounts for COPD so that they then develop symptoms and then progressive loss of lung function and associated diseases. So this acceleration of aging, we believe, is linked to a loss of endogenous anti-aging molecules. And what this means is that cellular senescence occurs in COPD, and there's a gradual accumulation of senescent cells. So a lot is known about cellular senescence because it's important for other important age-related diseases such as Alzheimer's disease, cardiovascular disease, and type 2 diabetes. And senescent cells are cells that have stopped dividing, and it used to be thought that they'd completely lost their function and they'd gone into cell cycle arrest. But what we now know is that cells in cell cycle arrest are actually metabolically active, and they produce a lot of inflammatory proteins. And this is called senescence-associated secretory phenotype. And it's something that we see in COPD lungs, an accumulation of senescent cells that include airway epithelial cells that lie in the airways, fibroblasts that surround the airways, or cells that make up the lung parenchyma. And these senescent cells are therefore producing multiple inflammatory proteins, which are exactly the same inflammatory proteins that are found in COPD patients, which makes us think that this is a major driving mechanism in COPD. We think the acceleration of aging and the induction of cellular senescence is driven by oxidative stress, which is increased reactive oxygen species that are found in cigarette smoke and wood smoke, for example. So this then drives the senescence, leading to inflammation, and these senescent cells are unable to, they lose their ability to repair. So it means that this is a progressive process. Now, what we've been working on is the pathways of senescence, which are well defined in other senescent diseases. And what happens is that oxidative stress activates a pathway called the PI3 kinase mTOR signaling pathway, which results in cell cycle arrest. And what we've shown is that this pathway also impairs key anti-aging molecules known as sirtuins. And the key sirtuins that are reduced in COPD are sirtuin 1 and sirtuin 6. And these sirtuins are known to protect against the development of aging and cellular senescence. So their loss in COPD is probably very important as a disease mechanism. There are several potential ways of reversing this aging process that we're currently exploring. And this involves inhibiting the PI3 kinase mTOR signaling pathway. And there are already drugs that are used for other purposes that can do this, including rapamycin, which is used as an immunosuppressant, and metformin, which is used as an anti-diabetic drug. So these drugs may be repurposed in the future to reduce accelerated aging pathways. There are also drugs that increase sirtuins, and one such drug is resveratrol, which is found in red wine. And it's been shown that many of these pathways that lead to accelerated aging are interactive. So this is a complex process, but it's possible that we can intervene with one of these new treatments. And these treatments have already been shown to prolong the lifespan of various species, from yeast right up to mammals. So we believe that this pathway will also be important in human aging diseases such as COPD. One of the areas that we've explored particularly recently is the role of microRNAs in regulating these aging pathways. And we've identified one particular microRNA called 34A, which is a key regulator of sirtuin 1 and sirtuin 6. And we've shown that this microRNA 34A is increased in COPD cells, and we can block it with a complementary sequence called an antagomer, and this is able to actually reverse cellular senescence in COPD cells. So the exciting possibility is that in the future we might even be able to reverse the aging process in COPD. Now, COPD is often associated with several comorbidities, and many of these comorbidities are also diseases of accelerated aging. And I think what's now emerging is that all these aging diseases share a common pathway. So they all have PI3 kinase mTOR signaling and microRNAs that may regulate this process. So what we believe is happening is that these diseases are occurring together as part of a multimorbidity. And multimorbidity is one of the key features of aging. For example, if you look at people over 60 years old, more than 65% have 2 or more chronic diseases of aging. So we think that these diseases occur together because they share common pathways. And this is very exciting for the future because it means that we may be able to develop treatments that deal with all of these diseases that occur together with a single therapeutic approach, which means rather than treating the individual diseases like heart failure, type 2 diabetes and COPD, that we may be able to use a single intervention that actually stops all of them from progressing and may even reverse these diseases. Multimorbidity is a major clinical problem because people are now living longer because they're not dying of infectious diseases. So this is something already happening in developed countries and increasingly in developing countries. And multimorbidity is the major challenge for medicine in the future because patients are living longer. A third of the babies born in the Western world now will live to be 100. And as people live longer, they develop more age-related diseases. So they develop several of these diseases together. And this is a major challenge for society. Harvard School of Public Health has estimated that multimorbidity will be the greatest single expenditure in the world over the next few decades. So research in this area, and COPD is a very good example of a disease affected by multimorbidity, is really important for finding ways around this treatment, and that includes not only drug therapies, which I've already discussed, but also changing lifestyle, changing diet and changing physical activity, which are all known to have the potential for reducing the accelerated aging process.
We are very interested in the aging of mammals because human is mammal, but also because mammals represent excellent experimental systems to study aging. So consider this, mammals differ more than 100-fold in lifespan. If you compare short-lived and long, like a shrew, which is short-lived, and whale, like a long-lived mammal. They differ more than 100 million-fold in body mass. And if we consider how this changes across evolutionary tree, branches of life, then it's very clear that nature has adjusted lifespan, or adjusting lifespan all the time. When it's needed, lifespan is decreased, like for example in mice and rats. When it's needed, it may be increased, for example, like in humans. So this process is reversible. So what puzzled us is the nature of this process, of how it can be naturally changed. This will be different in comparison with the experiments in the lab in which genes are manipulated and animals may live long, but these animals will not be competitive in a natural setting. We are interested in the natural changes in lifespan, which would still keep the species competitive. This area is not very well explored, and it's really just in the beginning of analysis. And our approach to this has been to understand the genomics of long-lived organisms. The first organism we've been becoming interested is called naked mole rat. It's a small rodent which lives in Africa, in the area of Ethiopia. It's underground in the subterranean niche. And it's the size of a mouse, but lives about 10 times longer, lives about 30 years. It's quite remarkable how it could be adapted to live that long. And of course it lives underground, it doesn't have enemies, the environment is quite stable and so on. But it has so many unique features. Besides longevity, it doesn't feel certain types of pain. For example, if you drop an acid on the skin of that animal, it doesn't feel a pain. It doesn't control the body temperature. It's actually the only mammal known which goes with the ambient temperature. So it's also one of the two known eusocial mammals. So they live in a colony with one female called the queen controlling the entire colony. That's the only reproducing female, and there are also 3 or 4 males which can reproduce. The other animals in the colony are workers. They never kind of reach sexual maturity, and they never reproduce. So those are quite remarkable features of the animal. But of course, for us, it's the most interesting longevity. So we became interested in this and decided to sequence and genome. And we had a nice collaboration with several labs, Shirley Buffenstein in San Antonio, Thomas Park at the University of Illinois, and the genome was sequenced at BGI in China, and also there was a nice collaboration with Eva Women's University in Korea, but we coordinated all this from here in the lab in Boston. So we sequenced the genome, analyzed it in various ways, looking for genes which are positively selected, looking for genes which changed expression in comparison to the short-lived organisms, looking for unique amino acids, looking for particular pseudogenes and other genes. And so we have various hypotheses with regard to how lifespan is adapted in naked mole rat. So one interesting lead is a protein called UCP1. It's a mitochondrial protein which controls the flux of protons going back to the mitochondria. It's called uncoupling protein, and the protein involved in thermogenesis. It's quite remarkable that that particular protein uniquely changed in naked mole rat, and naked mole rat does not maintain stable body temperature. So, and of course, if animals don't need to invest energy to produce heat, it's possible that they generate less damage, and this is one of the reasons they live longer. Some other interesting leads is one protein called P16. It's the most disease-related gene in mammals, and that protein is uniquely changed in naked mole rats, became shorter. So we don't know exactly how it affects the function of the protein. This research is still being done in the lab, but it's quite remarkable that this is a unique change in the naked mole rat. So there are various other features of the naked mole rat that we found. For example, 2 genes which are involved in telomere function, which is a process which has been implicated in the control of lifespan and aging, also uniquely changed in the naked mole rat. So we still actively study the animal and the genomics of the animal. And currently we are sequencing another genome of the mole rat, another mole rat called Damaraland mole rat, which is related to the naked mole rat, but it's like a normal animal. So this will help us to really define which features of the naked mole rat are linked to longevity and which features are linked to other unique traits of that remarkable animal. Another genome project that we have is on the bat, called Brandt's bat. This is another quite remarkable animal. It's perhaps even more extreme than the naked mole rat. So this animal weighs about 5 grams, but it lives more than 40 years. And it also flies, which is a metabolically active process. So apparently nature was able to adjust something in that animal so that it lives extremely long. So we just sequenced the genome of that animal and again looked for various genomic signatures, transcriptomic signatures and other characteristics which would help us to understand the exceptional lifespan of the Brandt's bat. So one interesting feature we found is the IGF-1 growth hormone axis. Growth hormone and IGF-1 signaling is one of the best known processes that regulate lifespan in model organisms. So for example, when a growth hormone receptor is downregulated, many organisms live long. And even within mammals, in mice, for example, the growth hormone receptor knockout mice, they are also quite long-lived. Interestingly, those growth hormone receptor knockout mice, they're also small. They're called dwarf mice. And here, as we have another analogy with the Brandt's bat, because bats are also small. So perhaps they have this dwarfism-like phenotype in which they adjusted IGF-1 and growth hormone signaling, so they became smaller, but this also made them long-lived. So we don't know the answer for this question, but it's still been an active area of research in my lab. We are interested in finding common features of various mammals. We think that some of the strategies in mammals, strategies to extend lifespan, are common. So it seems that some of the strategies involve central metabolism. It seems that the rate of metabolism decreases with the increase in lifespan. But this is probably one feature, and we are interested in identifying other common features. To understand that, we have to characterize many long-lived organisms. But in addition to the common strategies, it's apparent from these studies of the naked mole rat and the Brand's bat is that there are also species-specific or lineage-specific strategies, which each animal has its own strategy. And probably extended lifespan would involve a combination of both common strategy and species-specific. One challenge is to find signatures in the genome which are responsible for long lifespan. I mean, genomes are very large, and each species, they evolve according to habitat, the diet or environment and predators and so on. So because there are many changes in the genome, it's not easy to find features which are responsible for long lifespan and distinguish them from other features. So therefore, one approach to this problem is to study pairs of organisms which are closely related to each other, but one of them would be long-lived, one short-lived. And so for the naked mole rat, such a pair would be the Maryland mole rat, which is not really short-lived, but shorter-lived than the naked mole rat, but it's a kind of normal animal. Same thing for the Brand's bat. There are some bats, like an evening bat, which lives only about 10 years or so, but quite similar in terms of phylogeny. So with other organisms, like in some primates as well, there are shorter-lived and longer-lived organisms. So by studying those pairs, we can help to narrow down the genomic features which are responsible for long lifespan.